Well, we learned about how ATP is made, so now let's look at how proteins are made inside our cells. And please watch the video, that link that I provided for you. Alright, so information to make proteins is stored in our DNA. So really the job of DNA is to hold that information for making proteins. That's really it. And so then proteins determine what we look like. Okay, so let's go over here. The DNA is made up of these smaller molecules called nucleotides. And so many of these nucleotides come together to make up DNA and RNA. So it's not just for DNA, but it's also RNA. And so a nucleotide has this sugar called deoxyribose sugar. In RNA, it's the ribose sugar. Then there's also a phosphate group and also a molecule, a smaller molecule called a base. And so this base, so they wrote here B, this base can be an adenine molecule, a cytosine molecule, guanine, or thymine. So I'll show you on the picture next. So DNA has the two strands, and on the inside you have the bases. So if you look at the picture, we can see on this picture you have the DNA. And so the bases are in the middle. G and C go together, T and A go together, and the sides are the sugars and phosphates. And the hydrogen bonds are holding the bases together. Okay, so there isn't really much from this slide. Okay, so here comes the important stuff. Protein synthesis. Know that these are the two main steps. There's transcription and there's translation. Know that transcription is when mRNA is made from DNA. RNA polymerase is the enzyme that makes mRNA during this step. And then translation uses the mRNA that was made. And then there's also tRNA molecules and ribosomes to make proteins. So transcription, you're making mRNA. When making mRNA, DNA is opened up by this enzyme. So here, this big pink part is the enzyme called the RNA polymerase. It goes and sits on the DNA and opens it up. So when it opens it up, those bases are now visible. And there are inside, and also I should write down here, this occurs in the nucleus, which is where the DNA is. Nowhere else is where this is going to occur. Inside our nucleus are these free or smaller RNA nucleotides. Okay, so when those bases are opened up on the DNA, the RNA polymerase grabs those free nucleotides and attaches them to the DNA first. With that, as it moves along the DNA, that mRNA that is made is now coming out. And then once RNA polymerase does its job of making the mRNA, the DNA closes back up and RNA leaves. But again, the important thing is mRNA is being made in this step. And I wanted to show you here, if the base on DNA is A, then the base on mRNA is going to turn to U. If there is a C, it goes with G, G goes with C, T goes with A. Okay, so now next, translation. So with translation, we're using the mRNA, the tRNAs, and the ribosomes. Transcription occurred in the cytoplasm. Translation occurs in the cytoplasm. Transcription occurred in the nucleus. Sorry. So here, mRNA that was made leaves the nucleus and attaches to a ribosome. So then what we're going to see is the bases on the mRNA, each 3 base is called a codon. And then the tRNAs also have 3 bases. Their 3 bases are called anticodons. So this is just showing the different codons. You can see here, if this is what is on the mRNA, if there's a UUU, then the amino acid that it will translate into is called phenylalanine. If it's a UUC, it's also phenylalanine, but if it's a UUA, UUG, then it turns into leucine, so you get the point. And then if there are these codons, UGA, UAA, UAG, then the protein synthesis stops. Okay, so here. And also, I'm not sure why on this picture the little A is missing. But what we're seeing here, this is the mRNA. This is the smaller part of the ribosome, just a smaller piece there. The mRNA sits on the small ribosomal subunits. Only if there's an AUG, this is where the protein synthesis starts. Okay, so the first step, mRNA sits there on the ribosome, so now it's waiting. Then there are these other molecules called the tRNAs. So now this whole thing is a tRNA. tRNA has an amino acid at the top, and it has 3 bases at the bottom. Each 3 base on it is called the anticodon, and each 3 base on the mRNA is called a codon. Okay, so I always write this, and students see mistakes on this on the test by students. Codons are on the mRNA, and the anticodons are on the tRNA. Right, so UAC on this tRNA matches with the AUG. And then what it's saying is, then another tRNA comes in. So now, again, the codon and the anticodon has to match. But so when each tRNA comes in, each one brings an amino acid. So here on the next slide, we see how the amino acids are getting attached to each other. And so here the first one gets attached to the second one. So now here another tRNA comes in, brings its own amino acid. And at the end, all these amino acids attach to each other. When the protein is formed, when it's done, so then all these amino acids come out. And so polypeptide just means a protein made up of many amino acids. The start codon is AUG. You might see this on the exam. You don't have to memorize the stop codons. And so at the beginning, only AUG is being started at, and then the other codons are translated. Alright, changes in genetic information can occur through mutations. Some mutations can be beneficial or harmless, but other mutations can be very dangerous. And so, 2 types of mutations, they can be spontaneous. So sometimes DNA can go through changes like this just on its own. It can be insertion at the base. We don't quite understand why. Induced mutations are caused by mutagens. Mutagens meaning chemicals or radiation. Okay, so know the difference. Know that induced is actually getting caused by an outside stimulus. So some mutations are not harmful. Different eye colors, for example, or skin colors, were all because of mutations. So green eye color, blue eye color, it's just really mutations. But many mutations do affect our health. So changing even one base would change an amino acid, and if one amino acid is off, then the protein is not going to work. Okay, let's look at the next one. But thankfully, in our cells, we have these molecules that actually repair the DNA. It's called the repair enzyme. But it can still miss some mistakes. It's not going to catch all the mistakes, but at least we do have something like that.